Welcome to Study Loop.
Here we are talking about different factors of post-transcriptional regulation or control. So as the word indicates, this is going to be anything that occurs after the transcription of the mRNA. So you can imagine that you have your DNA strand with the gene of interest, and your polymerases will cause the creation of what we call the pre-mRNA strand. That pre-mRNA is going to undergo processing inside the nucleus in order to be converted into the finalized mRNA strand. And that, of course, can go through the process of translation in the ribosomes in order to create your protein. So here, when we talk about this post-transcriptional regulation, we are talking about this processing step to change the pre-mRNA to the mRNA strand. So that's going to include a few modifications. It's going to include the addition of a cap to one end of the RNA and the addition of a poly-A tail. And those are going to allow it to survive better in the cytoplasm where you have digestive enzymes that we would call RNases. So these poly-A tail and caps would prevent the RNases from digesting the important end of the mRNA strand. And the cap allows it to be recognized in the ribosome as well. In addition to those, processing is also going to include the process of alternative splicing. And alternative splicing is where we are going to cut out certain sections of the mRNA. So you can imagine that your mRNA includes several different gene groups. So alternative splicing can split out certain genes within there, and it's going to ligate the two ends together. And from there, you'll get a different product which will produce a different protein based on what's needed in the cell. So here, the post-transcriptional regulation is going to include the processes of alternative splicing and the addition of the cap and poly-A tail to the mRNA. For the other answer choices, you have the ubiquitation and you have the proteolytic digestion. And these are processes that are going to modify the protein strand with the addition of a ubiquinone group, which will allow it to be recognized for destruction in the proteasome. So those are more regulations of the protein rather than of the mRNA strand. So you can eliminate those answer choices because they are not post-transcriptional, because they're working on the protein itself. As for phosphorylation, this is the addition of a phosphate group to a protein, and that's typically going to activate, or in some cases, it can deactivate the protein. But again, that's working on the protein molecule rather than the mRNA itself. So here, the only post-transcriptional process is going to be this control of alternative splicing. So here, you should pick answer choice A.